Hello, everyone, and welcome to today's webinar, the 3-Peak Challenge for Long-Read Ultra-Deep Stool Metagenomics on the PromethION. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Zymo Research. Zymo Research is a privately owned company that has been proudly serving the scientific community with state-of-the-art molecular biology tools since 1994. The beauty of science is to make things simple is a mantra that is reflected in every Zymo Research product, from epigenetics to DNA-RNA purification technologies. Historically recognized as the leaders in epigenetics, today Zymo Research is breaking boundaries with innovative solutions for microbiome measurements. Zymo Research is the first company to develop microbiomics solutions from collection to conclusion based on new rigorous standards for microbiome measurements. Zymo Research has been leading the initiative to improve reproducibility and accuracy in the field by developing the first commercially available microbiome standard. To learn more about our sponsor, please visit www.zymoresearch.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education's Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Dr. Joshua Quick, Research Fellow at the University of Birmingham in the UK, and Dr. Janina Krumbeck, the Director of Microbiome Applications at Zymo Research Corporation. For the complete biography on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Hello, my name is Dr. Joshua Quick, and I work at the University of Birmingham in the United Kingdom. And today I'm going to present to you a talk titled The 3 Peaks Challenge for Long-Read Ultra-Deep Stool Metagenomics on the PromethION. So, nanopore sequencing is a relatively new technology. It was brought to market only in 2015, and it's notably a long-read sequencing technology. It's the second of 2 long-read sequencing technologies after PacBio sequencing, and what that means is that you're able to sequence very long molecules, which are important for things like de novo assembly. Up until now, the yields from these instruments has been quite limited, but with the introduction of the PromethION in the last couple of years, the opportunity has arisen to generate very large long-read data sets, and that's exciting because it allows you to increase the understanding of complex microbial communities, and this is generally known, an area known as metagenomics. So we particularly have 2 clinical translational projects here in Birmingham that we're interested in applying this technology to. The first is fecal microbiome transplants, which are being used more and more for the treatment of chronic C. diff infection. Our particular study is looking at the effectiveness of fecal transplants in a different condition called ulcerative colitis. And in that particular project, we're looking at the microbial community of the human gut before and after transplantation. And we have a second project where we're looking at the respiratory microbiome of cystic fibrosis patients. And this is a chronic lung condition, and it evolves over time. And we're interested in looking at the evolution of the respiratory microbiome in the disease. So the particular detail of the things that we're trying to do is that we're trying to enable single contig de novo assembly, and that basically means taking a very complicated mixture such as the human gut and using long reads, being able to de novo assemble short entire bacterial genomes from that complex mixture. And this is where it comes back to the fact that this has only really been possible with the introduction of the PromethION and being able to generate very large data sets of long reads, because the human gut contains many hundreds of individual bacterial species at a quite wide range of abundance, and in order to be able to assemble whole genomes from those, you need to produce sufficient coverage from those organisms to be able to assemble them. So we're trying to be able to link genes to chromosomes. We're trying to be able to identify strain level variation using haplotyping in the case of things like the evolution of microbes in chronic colonization in diseases like cystic fibrosis. But very specifically, what we want to do is to be able to track strains from before and after transplantation of fecal transplants. So we want to be able to see if individual strains persist after a patient has received a transplantation, and for that application, de novo assembly is very important to be able to assemble those genes and look at that variation. So we've been working on ultra long read sequencing methods for a very long time in Birmingham, pretty much since the introduction of nanopore sequencing. And ultra long read sequences are defined as reads over 100,000 base pairs, is our definition. So long read sequencing typically is classed as anything above 10,000 bases, and then ultra long would be 100,000 bases. The requirement in order to achieve ultra long read sequencing methods, you need very high molecular weight input DNA. And the way that we originally did this, we actually did the human genome with ultra long reads 2 or 3 years ago. And the way that we did that was using a phenylchloroform extraction method, which is very traditional, originally invented, I think, first published in something like the 1956, so very old method. And this particular protocol, which I used, came from a very famous book called Molecular Cloning by Sambrook, which many people will have seen if they've been in a lab. But basically, it relies on the detergent-based lysis of cells, digestion of proteins by proteinase K, and then removal of proteins using phenol emulsions before ethanol precipitation. And the way that we were able to generate reads over 100,000 base pairs, up to 1 million base pairs, was using a kit that Oxford Nanopore developed called the Rapid Kit. And that is, the kit relies on a method called tagmentation, which is done by using something called a transposase complex, and that is a protein which complexes with an oligo in a wild type. This would be a transposon, which is it is used. It facilitates the movement of genes, transposons around the genome. In this particular case, you use transposon ends, and you can sort of cut and paste the adapters into a genome, and it's very useful for producing next generation sequencing libraries. And the way that we managed to generate ultra long reads was purely by taking the standard kit and then putting a very large amount of high molecular weight DNA in there to minimize the number of insertions per molecule, so basically saturating the number of transposase complexes. Our first attempt doing this produced a data set on E. coli, which had an N50, and N50 is a measure that a measure of length of a read set of 65,000 base pairs with a maximum of 778,000 base pairs. So this is clearly something which is possible with cutting edge technology such as nanopore sequencing. Being able to thread a molecule nearly 1 million base pairs through a pore is seriously impressive stuff. So from the pulse field gel you can see on the right-hand side of this slide, we're able to extract DNA of 200,000 to 300,000 base pairs using this phenylchloroform method from E. coli. And that means that even if we have to cut or insert using the transposase, we can still get reads over over 100,000 base pairs very easily. So it's very suitable for what we were trying to do back then. The DNA input allowed that insertion and still generate long reads. The reason why we chose this kit was actually because it minimizes the amount of handling of the DNA, which we found to be the most important factor in generating ultra-long reads. And this slide here is actually the E. coli genome represented across the figure, and showing 8 nanopore reads which span the entire genome with overlaps, with an average length of 750,000 base pairs per read. So a tiling path across the entire 4.5 million base pair E. coli genome from 8 reads, and we're able to assemble these reads on a single core in 1.5 seconds. And this is really to demonstrate the power of ultra-long reads to be able to assemble genomes rapidly, and also to show that if you can get long reads, then de novo assembly is a completely trivial process, whereby all you're trying to do is identify 8 overlaps. This is a contrived example, really, but just to illustrate the point of why getting long reads is important, and why it's important is particularly in complex as mixes become more complicated, and the de novo assembly challenge becomes more complicated. For human stool extractions, for doing microbiome studies, we're not able to extract DNA anywhere near to 200,000 to 300,000 base pairs, and that has to do with the fact that microbes, in real clinical samples they're complex samples containing a mixture of viruses, bacteria, and fungi. They have very hard cell walls, some of them, compared to the human and the E. coli that we were demonstrating this method on before. So instead of trying to target reads of 200,000 to 300,000 base pairs, we're really targeting DNA input of 1 to 50 kb. So the other library prep method, which is common for nanopore sequencing ligation libraries, comes back into play, because we don't want to, if we're only able to extract DNA up to 50,000 base pairs, and we were trying to maximize our read length, we don't want to create an insertion site in the genome and fragment it more using a transposase. So we go back to the traditional method of ligation libraries, which originally we avoided, because what we noticed was that during the bead cleanup using Ampere XP beads, there was quite a high degree of fragmentation, and that resulted in, even if you had a high molecular weight input, that resulted in quite short fragments being due to shearing, basically, during the cleanup. But if you're starting with a shorter input, and there's also the additional fact that we've now managed to come up with ways to avoid doing bead cleanups at all, which was pioneered by someone called John Tyson in Vancouver, who discovered a way of using PEG and salt precipitation to avoid the use of Ampere beads. So Ampere beads actually work on PEG and salt, on a PEG and salt precipitation principle anyway, but instead of using the solid phase of a bead, he's using precipitation by spinning in a centrifuge. So he was able to produce a ligation library without any beads at all, and found that he could really maximize the read length from that method. So our idea was that we could apply ligation libraries and try to generate as high molecular weight DNA from stool as possible. We could achieve high yields on the PromethION. So that's what we set about to do. Now, it's very important when you're doing any kind of microbiome research to have standards for validation, and that's not just validation that your method has worked, but also can be important in development of extraction methods, but also downstream analysis methods such as assembly and taxonomic classification. And these are commonly called mock communities. So the mock community that we chose was a commercially available product called the Microbial Community Standard, and that comes in two types: even and log distributed. So the even contains an even representation of 8 bacteria and even representation of 2 fungi. The log is more to simulate a real microbiome sample where there's a large degree of difference between the most and the least and the most abundant organisms in the mixture. So the log distribution contains cells from 10 to the 2 to 10 to the 8, so over 6 logs of difference. As I mentioned briefly earlier, it's important to know that one of the big factors success of an extraction procedure is whether it works on gram-negative, gram-positive, and fungi. And in order to prove that, you need to have mixture you need to have a mixture of these different organisms in your validation sample. So this one contains appropriately a few gram-negatives, some gram-positives, and also some fungi. And the actual species are Pseudomonas aeruginosa, E. coli, Salmonella, this Lactobacillus fermentum, Enterococcus faecalis, Staph aureus, Listeria monocytogenes, Bacillus subtilis, and then the 2 yeasts are Saccharomyces cerevisiae and Cryptococcus neoformans. So it's a good range of organisms in there. And it's actually classified as cellular, so intact cells, except the fact that in order to inactivate it for shipping, it's shipped in something called DNA and RNA, DNA/RNA shield, which actually does cause some lysis, especially in the gram-negative organisms. So when we do this, we make sure that we retain the supernatant, if you like. So if you were to pellet it out, you would pellet some cells, but also it's important to retain the supernatant because that will contain some DNA from gram-negative organisms that have lysed. So yes, and there's also Illumina data available for the community. So we've recently published our results using the microbial standard, both the even and the log, and sequencing on the Oxford Nanopore PromethION instrument. And this is actually the first peer-reviewed publication which contains data produced by the PromethION instruments, which is it's very cutting edge. And these are some basic run statistics. So the plot A shows a collector's curve of data generated. So the log and the even community on the PromethION generated in excess of 160 gigabases in a 90-hour run or longer. The 2 lower down curves show the MinION it's actually the GridION flow cells, which we did, and you can see that they generate about 15 to 20 gigabases per run. The figure on the right shows the even and the log communities for both the GridION and the PromethION, and you can see in terms of accuracy, the modal accuracy of the instruments is in excess of 90% and very consistent between both the 2 nanopore platforms and both the 2 communities of the even and the log. So it's all so that's something that we're able to is a validation that we're able to perform. From the log community, we can also produce alignment statistics for each of the organisms against the reference genome for both the PromethION and the GridION, and you can see that the diagonal line represents the expected abundance of those organisms, and you can see that from the alignment statistics, they fall very nicely and tightly along the diagonal line, which means basically that we're sequencing the expected proportions that we're hoping to, and that tells you that you're not introducing any biases in either the extraction or the sequencing or the analysis procedure. And in this particular case, we're doing bead beating, which is on the entire sample, so you wouldn't expect there to be very much bias. This is a table showing the same data, and you can for the log community, and you can see that for the highest abundant organism in the log community, the Listeria monocytogenes, we've got 36,800x coverage for the PromethION data, and then for the next 6 organisms, we have 20x coverage, which we would term to be sufficient to assemble the genome. And then below that, we have 2x coverage for the Lactobacillus fermentum, which is enough data to identify genes, a subset of the genes, and some genome scale information, but not enough to assemble. And then below that, for the Faecalis, the Neoformans, and the Aureus, we have detectable quantities, but very, very low, not enough to find very much information out about the organism, but just enough to detect it. So you can see that even with the vast sequencing power of the PromethION, we're still able to be stumped by this 6 logs of abundance. So this really represents the sort of state of the art at the moment, and it works over a fairly wide range. But we're only able to detect the lowest input, which was the Staph aureus, which represented about 400 cells in the sequencing library, which was only 1 picogram. So we are able to detect all the organisms, 6 out of 10 organisms at high enough coverage to assemble, 2 to preserve some genome scale information, and 1 to preserve some to provide some genome scale information, and then 3 to provide just to be detected. So the next section is about using the community to optimize our extraction methods for increased read length. And the first section, the first slide I'm going to present here is maybe a little bit controversial, but I think it's important to note that what I did in this experiment was I took the microbial community and I used the same lysis procedure, but then I took the lysate and I split it between a spin column and a magnetic bead extraction, and then sequenced them both. And what you see here is that the spin column causes so much shearing down to 10,000 base pairs, the sort of maximum that you can get from the spin column extraction. That even though you have this potential, these long fragments in your lysate, there's so much shearing caused by the spin column procedure that you're limiting your read length to about 10,000 base pairs. So I make the slightly bold statement here that spin columns do not support long reads in this particular application. And I would recommend the use of magnetic bead base cleanup in this case, which will allow you to recover up to 30,000 base pair DNA fragments. And this is a very able to make a very sort of tightly controlled comparison here, due to the fact that Zymo make these 2 kits, the mag bead kit and the spin column kit, with the same binding chemistry. So these beads are silica beads, and they use the same binding chemistry as the spin column. So you're able to split the lysate, and it's a very tightly controlled experiment. So I've already briefly talked about the differences in the cell wall compositions of different organisms, but this really is the crux of the problem, which is that you get a very wide diversity of bacterial species in your real clinical samples, and they have very different compositions. So bacteria, gram-positive bacteria, have a tough cell wall. But both gram-positive and gram-negative bacteria can also have an additional capsule layer. Then, if you're looking at a microbial community, you're also going to have yeasts, which can have very tough beta-glucan or chitinous layers protecting them, and you can also have spore-forming bacteria. which can have a spore cortex of peptidoglycan, and also they can have a keratin coat. So these very, very stable, difficult molecules to disrupt. In addition, and as well as that, they also have a very wide range of chemical compositions, which make digestion quite a challenging procedure. So to tackle this, there's Scott Tai, who should take most of the credit for this work, has developed something called Metapolyzyme for the ABRF Metagenomics Research Group, which is a cocktail of 6 enzymes. You might have heard of DNA extraction enzymes in terms of the sort of common ones such as lysozyme, but what he did was cocktail together 6 enzymes: metanolysin, chromopeptidase, liticase, chitinase, lysostaphin, and lysozyme together in a single buffer where they all have activity. And what that will do is sort of attack the broadest possible spectrum of chemical compositions of cell walls in bacteria and yeast. And what that will do is digest away the cell wall, leaving only the cell or the plasma membrane bound cell, which is either a seroplast or a protoplast in gram-negative or gram-positive organisms. And then that leaves it to be very susceptible to a chemical lysis by detergent. So we can avoid sort of shearing physical disruptions such as bead beating in favour of gentle enzymatic digestion in order to recover longer fragments of DNA. And that's really the basis behind our method, which we call the 3 peaks method. So when we take a real sample, or the mock community, or a real sample, we expose it to 3 sequential methods of chemical, enzymatic, and physical lysis, increasing harshness effectively. And what we do is we remove the DNA that's be able to recover at each individual step, so it doesn't get damaged by the subsequent step. So, for example, after chemical lysis, we spin down the remaining cells, take off the supernatant, put it to one side. Then we take all the remaining cells, we expose them to an enzymatic digestion with Metapolyzyme. Then we spin that down, and anything that's still any cells that are still remaining at the end, we expose to physical disruption of bead beating, which is going to cause quite severe DNA fragmentation, but it will maintain the representation. That's the crucial thing. You can't trade off your fragment length for your representation, because you have to know what's in your sample, even if you can't recover high molecular weight DNA from it. So this is an experiment here where we show the abundances of the organisms in the mock community with different treatments. So the red is the supernatant and the bead beating. The green is the supernatant and the Metapolyzyme treatment, and then the blue is the supernatant, the Metapolyzyme, and the bead beating, the full 3 peaks method. And what you can see notably is that without the bead beating, we lose the representation of the Cryptococcus. So that the Cryptococcus neoformans is very underrepresented, 25x times underrepresented when we don't do bead beating. And in this particular case, this is an excellent example of the validation data showing you that in this case, the strain of Cryptococcus neoformans in the mix was not susceptible to the digestion by the Metapolyzyme. And this here shows the optimized extraction method that we developed using the magnetic beads, but also the 3 peaks together, showing that we're able to sequence to produce DNA from the microbial community with an M50 of nearly 26,000 base pairs. So that's the combination of using the splitting out the extraction to preserve the fragment length with the 3 peaks, and then doing the cleanup isolation using the magnetic beads, which we found to be able to preserve DNA. So we call that the optimized method. And we also assembled these data sets, and this is done using a program called Fly, which is a sort of most recent generation single molecule assembler using a repeat graph algorithm. And to be honest, the assembler is so good that this is the non-optimized data. So this is showing the comparison between the original GigaScience dataset, which was just all bead beaten, and then the optimized method, which was the 3 peaks and the magnetic bead extraction. And really, the assembler is doing such a good job, even on the bead beating, that you don't see so much difference here with these bacterial species. But if we look at the yeast, obviously it's a larger genome containing, considerable numbers of repeats. You start to see here that the optimized method with the long fragments is generating larger contigs for the yeast species, particularly the Saccharomyces cerevisiae here. Now we don't have the data for all of the 10 species because we didn't have a reference genome for all of these. In order to produce this dot plot, we needed to be able to have a reference genome, so we have the Saccharomyces there. I'd also like to draw your attention to a video if you're interested in learning more about Fly and how the algorithm works. There's a video on the Long Read Club YouTube channel, which is youtube.com/longreadclub, and that's a good resource for that we're starting to compile information on long read sequencing. And now I'm going to show here a slide which contains all of our fecal extraction PromethION and MinION data that we have so far. And these are the real clinical samples. So they're the N50s here, they're a lot shorter than the 26,000 base pairs which we're able to achieve with the optimized method on the mock community. But still we were still able to generate N50s up to nearly 17,000 base pairs for some of these real clinical samples, which we're very happy with, because this is a real clinical sample containing a complex clinical sample, difficult to work with, real feces rather than just a cellular mock community, and we're still able to generate some really nice long reads with this method up to 17,000 base pairs. And you can see that the yields are also very high. Some of these PromethION runs, we have 2 in excess of 110 gigabases. So this is giving you the length and the yield required to assemble these and do some really fine detailed analysis of these communities. And to illustrate the 3 peaks method again, this is a histogram of the read lengths showing what the actual read lengths look like if you do this. So we do you can see from the histogram there is only really 2 peaks, and that's because the chemical lysis probably from the first step and the metapolyzyme, they both produce quite long DNA of 20 to 50 kilobases, and those distributions overlap, so they appear to be 1 on this plot. And then you see the bead beating, which is where you put the cells inside a tube containing a matrix of particles, which can be some mineral like garnet, things like that, and then you expose it to very high energy using a bead beating instrument, which is sort of reciprocates or figure eights the beads around the tube, causes that sort of physical disruption, that smashing open of the cells. And some of these instruments are extremely powerful. This is proper bead beating, using a fast prep or something like that, moves the sample at sort of 6 meters a second or something. It's a very large amount of energy that you're putting into that sample. In fact, if you bead beat a sample for about 1 minute, take the tube out, it's hot to the touch. That's how much friction these beads cause. So that's why you're able to preserve that representation, because you're really using a very large amount of energy to disrupt those cells. I just would like to say that for the US audience, the 3 Peaks Challenge, which this is named after, is actually a sort of United Kingdom centric outdoors challenge, which is actually to visit the largest, the tallest mountains in in England, Wales, and Scotland in the 24-hour period. So the future perspectives and sort of as a summary for this talk, read lengths are relatively short when using the conventional bead beating or column-based extraction methods. We've developed ways to avoid the pitfalls of bead beating and both column by changing to the 3 peaks, which has the chemical and the enzymatic lysis beforehand, and also moving to the mag bead-based extraction methods. And that's shown nice improvements in fragment lengths, which are going to produce better results when you do long-read sequencing with them. We found that metapolyzyme was an important component in this, being able to produce long DNA from all of the gram-positive organisms in the mock community, but also one of the yeast in the mock community. And we think that that's an important method going forward. There's actually going to be a metapolyzyme 2.0 in the pipeline, which we're going to be involved in the specking out and the production of. So optimized extraction can give very long reads on nanopore sequencing, and that converts into highly contiguous assemblies. And when the yields on PromethION are now sufficient, over 100 gigabases, for tackling complex communities such as the human gut microbiome project. The yields, however, the inputs required for PromethION flow are on the order of 500 to 1,000 nanograms, but you can produce that with sort of 100 milligrams of stool, and that's what we do. It's not a large amount. And that we're able to recover genome scale information over a range of 4 logs using with yields like this, and detection of organisms over 6 logs. And all of the data that I've produced, that I've talked about today, is all available on the online. This is all open data, open analysis. I work as a postdoc in Nick Lohman's lab. This is the GitHub for the Lohman lab, and all of the work here is under a page called the mock community. And then finally, I'd like to acknowledge my colleagues at the University of Birmingham and Sam Nichols, who did a lot of this work in terms of the analysis. Scott Ty, who's developed and helped us out a lot with the metapolyzyme work. John Tyson, who developed the bead-free method. Our contacts at Oxford Nanopore and Zymo Research, whose technologies I've mainly talked about today, and Hannah McDonald for providing the mock community. That's the Zymo distributor in the UK. And that's the end of my talk. Thank you for listening.
Well, hello, and thank you for joining us today for the Zymo Research webinar. My name is Dr. Janina Krumbeck, and I'm the Director of Microbiome Applications at Zymo Research. Today's topic is how we can explore the microbiome with confidence using microbiomics-grade tools and standards. Those tools can help improve the accuracy and reproducibility of microbiome measurements. You probably already know what the microbiome is. It's the study of microbial communities, the study of microbial ecosystems that live everywhere around us. And from the first idea of a living cell that was described by Robert Hooke in 1665, microbiologists have been discovering this whole world of microbes that surround us in an ever faster growing pace. From the first notion that microbes can ferment food and produce beer and cheese, to the understanding that bacteria cause diseases. Many of the techniques that were developed in the golden age of microbiology in the late 1800s are still being used today for medical diagnostics, such as gram staining or the Petri dish. But the biggest push for the field really came with the discovery of the DNA helix and the discovery of the 16S RNA marker. Suddenly, we found out there's this whole kingdom of life that we didn't know about before. Because most of the microbes cannot be grown in the lab, The sequencing technologies developed faster and faster, from the first bacterial genome being sequenced in 1995 to the Human Microbiome Project and Ocean Sampling Day. Now we know that there are about 10 to the 20 microbes on Earth and about 100 trillion microbes on each of us. However, 99% of those microbes are not culturable in the lab, yet they have a huge impact on our lives. The tools to study which microbes are present and what they are doing on a large, high-throughput scale are referred to as metagenomics and next-generation sequencing. This term, next-generation sequencing, or short NGS, can be a bit misleading, as NGS was actually developed about 15 years ago, and we are now in the era of third-generation sequencing. And with the development of NGS and decrease in sequencing cost and sequencing time, researchers started studying everything from the oceans to our pets, to ourselves, to agriculture, and even the highest points on Earth. Researchers have been so excited about the possibilities that NGS has offered them that the microbiomics field has grown nearly threefold by the numbers of citations since 2005, with more than 7,000 new publications per year. One of the leading scientists in the field is Dr. Chris Mason from Weill Cornell Medicine. He's one of Zymo's closest collaborators, and he's really doing amazing work analyzing the microbiome of cities that we live in tracing microbes as well as antibiotic resistances. And he says that it's indeed an exciting time for the field of metagenomics and the microbiome, but there's also limited ability to compare between different research studies, which has greatly hindered the progress of research. And what he means by that, I would like to illustrate in two examples. You might have heard about this story before, as it came out in 2012. A researcher collected her own fecal sample and split it into two, and then she sent the two samples to two major US companies that offered fecal microbiome analysis and compared the results, which are shown here on this slide. And the results were drastically different. Both profiles agree that the Bacteroidetes and Firmicutes are the dominant phyla in her sample, but American Gut showed that the abundance of Bacteroidetes is much higher than that of the Firmicutes, but the uBiome profile shows the complete opposite. The abundance of the Firmicutes is much higher. So just one single sample, two dramatically different microbiome profiles by two big players in the field. And what you need to know is that these both companies use very different protocols to process and analyze the sample. But still, we don't know which one is correct. And while that was just a single sample, this example highlights the drastic impact that different protocols have on the microbiome profile. These are some of the largest microbiome studies yet conducted, looking at the human fecal microbiome, the Human Microbiome Project on the left and MetaHead on the right. The Human Microbiome Project alone had funding of $200 million US, but just like before, the results are drastically different, even if we're looking at just high taxonomic levels like the phylum. And they, again, are reporting the opposite results regarding the ratio of the Bacteroidetes and Firmicutes. Again, both projects use different protocols, and we don't know which results are true. And when we take a more detailed look at the microbiomics workflow, it's not surprising that the results can be so drastically different when using different protocols, as each step of the workflow can have a huge impact on the final result. I will go over the major causes that introduce this bias to the microbiome measurements. The first major driver of bias is the sample collection and storage. Keep in mind that you are collecting living organisms that are highly diverse. Some organisms may decay or keep growing in your sample, and that's what I'm illustrating here. They're so diverse, it's like trying to store and ship dry ice cubes versus rocks versus bread. One may disappear really fast, others are quite stable, and others may start molding and decaying. And to illustrate what this means, even major publications like the American Gut Project struggle with things like this. Here's an example of a fecal sample, and it's not protected, and we're just looking at a higher phylum level, and you already see a big difference from day 0 to day 1, where there is this pink growth on top, and these are E. coli. And E. coli can drastically grow very fast in your sample. And like I said, even major publications face the challenge of sample collection and that bacterial growth and decay of nucleic acid degradation compromise microbiome measurements. In this case of the American gut, there was an increase of E. coli introduced by the sample collection, and it required editing of the data to retry and remove bacterial brooms of E. coli artificially from the data after the samples were already collected and processed. Those issues can be avoided though. Zymo Research has developed DNA-RNA preservative called DNA-RNA Shield that prevents this from happening and can preserve the DNA for up to 1 year at room temperature. So now that we know that we can store our samples without worrying that any changes to the microbiome occur, the next step is the DNA and RNA extraction. Again, we are dealing with microbes that are highly diverse with many diverse characteristics. Some are easy to lyse like E. coli, others are difficult to lyse like Listeria with different cell sizes. Just like this example, you might need different tools to break open an egg versus a marble. If you don't lyse the cells, the DNA of those organisms won't get sequenced, and the organisms are underrepresented in your samples. So depending on which lysis method you use, like bead beading or chemical lysis, the results are very different. This is one of the major reasons why the results from the Human Microbiome Project and MetaHit are so different, the overrepresentation of easy-to-lyse organisms. This is something you should particularly pay attention to when choosing your protocol. I don't have time to go into details today, but Zymo Research has also developed DNA and RNA extraction kits that can avoid this type of bias. Bias in the library prep can be introduced based on varying GC content, PCR inhibitors, the formation of chimera, or the primer design itself, which may not cover all organisms in your sample. The sequencing platforms themselves also have some sequencing errors associated with them. And finally, the bioinformatic pipelines that are using can have a huge impact as well, depending on how the alignments, the chimera removal, the clustering are handled, to name just a few of them, and of course, which reference database you're using. As Dr. Cano put it, relatively minor alterations in the DNA extraction procedure or in the bioinformatics analysis can give a distorted view. And none of these steps in the workflow are trivial, but the workflow is complex and prone to error. Many products and methods have not been validated for microbiome work simply because those tools were never designed for microbiome or NGS work. Many buffers for the extraction never needed to be as clean as we need them to be now, because sequencers can start picking up bacteria that were added to your samples simply because they were already present in the buffers. So this is very particular to ensure that your buffers are clean. With the rapid growth of the microbiome field, there has been consequences. The accuracy and reproducibility of issues are rampant, and we know about those issues. And this paper was published in 2015, but 4 years later, the results are still the same and the requirements are still the same. For every PCR that you run, you need a positive and a negative control, but for microbiomics measurements, there's currently no such requirement by any journals. And Costea et al. Did an extensive comparison between different studies, comparing 21 of the most cited different DNA extraction protocols. And here I'm listing some of their highlighted findings. They found that the protocol used dramatically influenced the conclusions taken from comparative studies. Every step from sample collection and storage to the extraction methods used had a huge impact on the outcome. So the variation between protocols was larger than the variation between subjects. And they're not the only ones that have been looking into this, and there are now several more studies that have been published and look specifically at this problem of bias between different studies and extraction protocols. But we as researchers rely on other publications to conduct and do our own research. For every grant proposal, every paper we write, we put our own research into reference what other people have been publishing. And this has great consequences on our own work. So let's just look at a very simplified example. Let's say study A claims we found a large gem, which is pretty amazing and everyone is excited. But then study B says, hey, we found a small gem. And when you start comparing those two studies, those two gems look pretty much alike. But is it now a large one or a small one? And how can we compare those results? Especially now that we know that every single step of the protocol can have a huge impact on the results. But how can we draw any conclusions without using a reference? Because measurements are always relative, but once we add a scale to the findings, we can actually start comparing the results. Of course, there are different kinds of scales that we could use, and some may be more appropriate than others. And even in the history of our world, we haven't quite been able to just agree on a single scale to measure something like distance. But ideally, we can agree on just one or two scales for microbiome measurements so we can translate the findings and communicate our results clearly with one another. Because keep in mind, every single step of the workflow influences the outcome and even change the perspective of the microbiome and the results we find. And I hope that I've convinced you that using a standard for microbiome studies is needed. Zymo Research has now developed multiple standards that can help you identify if there is any bias in your workflow, all the way from sample collection and preservation to the bioinformatic analysis. This is what Zymo's Microbial Community Standard #1 looks like. It's the mock community of 10 different organisms, 8 bacteria and 2 yeast in an even abundance. These organisms were specifically selected to represent a wide variety of different cell sizes, toughness of the cell wall, and GC content like you would find in a wide variety of different samples. This standard can be used for general benchmarking and microbiome profiling. The whole cell is the mock community for the complete workflow, so everything from sample collection, DNA extraction, and so on. And the DNA mock community is for the library prep and bioinformatics part of the pipeline. Of course, accuracy matters here. If you want to benchmark your work to a standard, that standard has to be as accurate as possible. Zymo Research guarantees that there is less than 15% deviation from the defined composition and less than 0.01 foreign microbial DNA contamination. The same standard is also available on a log scale to assess the detection limit and sensitivity of the workflow. Again, it's provided as a whole cell mix or as the DNA mix. Zymo has also developed spike-in controls. This standard can be used as an internal process control and enables absolute quantifications of microbes in a sample. So there is no need to run additional qPCRs, for example. The standard is available in high and low bacterial load, depending on the bioburden of the sample you're using. So you know exactly how many cells of these 2 organisms that we provide you're using, and you can see at the last step of your workflow how much % of those 2 organisms do you find, and you can calculate back the absolute abundances of your cells. So this is a really great tool that saves you a lot of time running without the need to run additional qPCRs. The new standard that Zymo has developed is the ZymoBIOMICS High Molecular Weight DNA Standard that was created to help validate long-read metagenomic tools and pipelines. It's composed of purified high molecular weight DNA, larger than 50 kb, from 7 bacteria and 1 yeast as listed here. The taxonomic diversity can help with the development and validation of long-read metagenomic bioinformatic tools, just like we heard Dr. Quick talk about earlier. As shown in this graph on the left, high-quality read DNA data is generated from the ZymoBIOMICS high molecular weight DNA standard. In the Oxford nanopore MinION sequencing run shown here, the histogram shows an average read length of 24 kb and more than 125 kb recorded. So Zymo Research is offering a control for the entire microbiomics workflow to address every single step that could introduce bias into your data, from sample collection and preservation using DNA-RNA shield to using specific DNA and RNA extraction methods based on your sample type and needs, to library preparation kits, and we also offer next-generation data sequencing analysis. So for the past 10 minutes, I've described the immense challenges faced by the field, but also showed you that they can be resolved. Zymo Research has built a complete pipeline for microbiome studies and has resolved major workflow challenges faced by customers. I didn't have time today to go into specific details, but Zymo is offering a solution for every step of the way, providing standard solutions for unbiased sample collection, unbiased DNA extraction, library preparation, and data analysis. We are currently offering the Microbiomics Standards and Controls Initiative, the MySci initiative. You can simply tell us about your project, and we will provide you with free standards that you can incorporate into your workflow to see if there is any bias in your workflow. You simply sign up on our website, and we will send you the mock community standards just as you need it. And with that, I'm at the end of my presentation, and I would be happy to answer any questions that you might have. Thank you.
Thank you, Dr. Quick and Dr. Krumbeck, for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question is, how can the read lengths be improved further?
I think that's a good question. Really, what we're talking about is how do you improve from read lengths of sort of tens of kbs to bridge the gap to what we're able to the ultra long reads, which we're able to achieve on certain gram negatives and human cells. And that is definitely not an easy task for many gram-positive spore and fungi. I think that really, the options we have really are further development of enzymatic approaches to broaden the substrate specificity of those, to move to some entirely novel form, or some entirely explore other sort of non-standard forms of cell lysis. There are some examples already of using heat cycling or freeze thaw for cracking cells open. There's examples of ultrasonication being used to disrupt cell membranes. So really, I think that unless there's some completely non-standard method of cell disruption, then really we're talking about further development or optimization of chemical and enzymatic methods.
Thank you. All right, the next question is, how will this help further understanding of human disease?
this question goes back to my introductory slide, really, and I gave a couple of examples of projects which we're involved in here at Birmingham. The first being the fecal transplantation clinical trial, and in the clinical trial we're collecting fecal samples from day one of the recipient, the receipt of the donation of the transplantation, and then at day 56 of the transplantation. And what we're interested is how many species persist to 56 days. And the way that we're doing that is by doing shotgun metagenomics, so extracting all the DNA from those samples, assembling them, and then trying to identify persistence of individual strains. And it's quite a sort of simple, basic analysis that you might have done in the past. Might have been to do some sort of 16S, which will give you a genus or potentially species level assignments, or Illumina short read metagenomics, which would potentially give you species level assignments. But really what we're talking about now, the cutting edge, is whole genomes and strains. And can we identify, strain level diversity within a species, within a microbial community? And that's really where the hot ticket is these days. In order to further our understanding of microbial communities, the dynamics at play and the interactions with human disease.
Thank you. It looks like we have time for one more question. Are there any other methods for cell lysis in development?
So I briefly mentioned a couple of other methods that I knew about in my answer to the previous question. So in addition to chemical, which are usually detergent-based lysis, detergent-based methods for solubilizing membranes, I also talked about enzymatic methods and the development of broader spectrum enzyme cocktails, such as metapolyzyme 2.0, and then I talked about physical disruption methods, which normally involve very harsh physical treatments, such as bead beating. Then I mentioned freeze-thaw processes, where you try to basically freeze the sample in order to crack the cell open via the action of the expansion of water, which obviously expands when it freezes, and also ultrasonication methods as a sort of non-standard approach towards disruption of microbial cells. I can think of a couple of other non-standard methods, such as cytotoxins, so pore-forming proteins, similar to nanopores themselves, actually, but in a completely different context. Any proteins which can puncture their way through cell membranes could be one possible way of causing cell lysis. Antimicrobial peptides, another class of molecules which could potentially be used in the same way. And that's all I can think of at the moment.
And your important research. We would also like to thank LabRoots and our sponsor, Zymo Research, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speakers via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.